While they're not talked about nearly as much as the nucleus or the mitochondria, ribosomes are easily one of the most important organelles in the cell. There are about 10,000 per cell, so you have about 4 quintillion in your body at any given time. Ribosomes are synthesized in the nucleolus and can be found in two major places in the cell. The first place is the rough endoplasmic reticulum. It's studded with ribosomes and tends to make proteins that attach to or leave through the cell membrane. The second place is the cytoplasm. These ribosomes are free-floating and usually make proteins that are used inside the cell. Ribosomes are a little more complicated than they look, but they're still fairly simple compared to most organelles. They're made up of two subunits, conveniently called the large and small subunits, and they have three internal tRNA docking sites called the A site, the P site, and the E site. Like I said before, ribosomes synthesize proteins, and they do this through translation. To translate mRNA, a ribosome binds to an mRNA strand and reads it in a three nucleotide sequence called a codon. There's 64 different codon combinations, which code for 20 different amino acids. This is an amino acid table, but you really won't have to worry about memorizing it. You'll almost always be provided with one. But the most important codons you do need to remember are the stop and start codons. Stop codons tell the ribosome to stop adding amino acids to the protein chain, but they don't code for any specific amino acids. Start codons, on the other hand, code for the amino acid methionine, and they initiate translation. And there's only one start codon, so every protein starts with methionine. Now that we understand what codons are, let's take a closer look at how ribosomes translate mRNA strands into proteins. Translation has 6 major steps, so we'll go through them one by one. The first step is binding to an mRNA strand. The A site always binds to the start codon, and the nucleotides face a large subunit. Since tRNA molecules are free-floating in the cytoplasm, there's almost always one floating nearby for the ribosome to use. The second step is to grab a free-floating tRNA molecule. tRNA molecules consist of an amino acid and an anticodon. The anticodon dictates which codon it binds to, and the amino acid it is donated at the P site. The third step is for the anticodon to line up with the start codon as the tRNA moves to the A site. The fourth step is for the ribosome to grab another tRNA molecule. It has to be complementary to the next codon in the strand, and it moves to the A site. At the same time, the tRNA that was in the A site moves to the P site. The fifth step is for the first tRNA to move to the E site and donate its amino acid to the tRNA in the P site. These 5 steps repeat, and the ribosome continues stringing amino acids together until it reaches a stop codon. The stop codon initiates the sixth and final step, which is for the ribosome to release both the mRNA strand and the protein. The protein folds by itself, and the ribosome is free to bind to new mRNA strands. Since we just went over a lot of information really quickly, here's a brief summary of translation. Ribosomes read 3 nucleotide sequences called codons. For each codon, a ribosome summons a free-floating tRNA molecule with a corresponding anticodon. Ribosomes take amino acids off the tRNA molecules to string them together to make proteins, and ribosomes aren't involved in the protein folding process.